Welcome in the part 8 video lecture of viral delivery system. So today we are going to discuss about lentiviral vector. So in our previous 3 video we have discussed about baculovirus based vector in very much details. So now we will going to discuss about lentivirus vector. Okay, so at I think 765, so part 4 we have discussed about retrovirus. So if you have not watched about what is retrovirus vector, then I will recommend you to go and first watch this one, then come back here, so that it will be helpful to you. Although I will give a quick recap of retrovirus also. So retrovirus is a positive sense RNA virus. It contains mainly single stranded RNA as their genome of near about 8 to 9 kb, so 7 to 11 kilobase pair it is there on at about length. Okay, so among them the insert size is 8 kb. Okay, so this retrovirus have the ability to transduce mainly dividing cells, because remember it rely on, or the transduction with the help of retrovirus rely on the breakdown of the nuclear membrane. Okay, and that is only possible during cell division, right? That means retrovirus can only transfect dividing cells. Okay, although it do not have a wide range of cell tropism, but we can construct a wide range of cell tropism with the use of a special type of envelope known as vesicular stomatitis virus glycoprotein. That envelope or that glycoprotein will make the retrovirus a viral vector that can infect any mammalian cell types. Okay, another important thing is that the major disadvantage of retroviral based vector is retroviral based vector have the ability to stimulate insertional mutagenesis. Okay, and also it have the ability to activate latent disease. Okay, and the most important advantage of retrovirus is the retrovirus gives you a stable expression, because remember in our detailed discussion of AIDS disease we have also discussed that the replication of the HIV virus or any kind of retrovirus, right? So here we have discussed that it will integrate with the host genome, right? So if it integrates with the host genome, we have discussed that it have the ability to facilitate the stable expression of the transgene. Okay, so that was the overall discussion of the retrovirus. So now we are going to talk about lentivirus. So lentivirus is nothing but a subgroup of the retrovirus itself. That is why I am telling you to go and watch retrovirus once again, then it will be helpful for you to understand. So that means lentivirus is again a single stranded RNA containing virus. So lentivirus, the main difference between lentivirus and the retrovirus is that lentivirus have the ability to transduce both dividing and non-dividing cells, whereas in retrovirus only capable of the transduction of only dividing cells, right? Now another important thing that I want to mention, that lentivirus also ensures a stable expression. Okay, because it will, as it is a subgroup of the retrovirus, it will also integrate with the host genome. Okay, and it transduce non-dividing cells and terminally differentiated cell population, for example neuronal or hematopoietic cells, okay, by a process called active nuclear import pathway. Okay, that is why lentivirus is considered as a more versatile tool among all the other kinds of retroviral vector available. Okay, so size of the genome as equal to the retrovirus, because it is a subgroup of retrovirus. So 10 kb, DNA insert size 9 kb, maximum titer 1 into 10 to the power 9 particles per milliliter. Okay, so these three parts are exactly same that we have discussed about retrovirus, more or less same, because in retrovirus we have discussed about 11 kb size, here 10, the DNA insert size is 8 kb in case of retrovirus, here 9, and maximum titer is same in both the cases. But the difference is in infection, because retrovirus was capable of only infecting dividing cells, right? But it has the ability to transfect non-dividing cell too, okay? And it transfers the transgene via active nuclear import pathway. Mind this very carefully. And expression stable because it can integrate with the host genome. And the drawbacks is, as like as retroviral phase vector, it has the ability to induce insertional mutagenesis, okay? So that is the drawback. So one more time, lentivirus. Lentivirus is what? Lentivirus is a subgroup of the retrovirus. It can also ensure a stable expression of the gene because it can integrate with the host genome. Okay, clear? Now, unlike other retrovirus, lentivirus only have the ability to transduce non-dividing and terminally differentiated cell types. For example, hematopoietic cell types or neuronal cell types, okay, via a process called active nuclear import pathway. Okay, so that is the idea. So it can transduce both dividing and non-dividing cells that retrovirus can't, because retrovirus only transfecting dividing cells. Okay, and another important thing it is that your lentivirus is transferring the transgene via active nuclear import pathway, but in case of the normal retrovirus there are not such kind of pathway. That is, among other retroviral base vector, lentivirus is most preferable. Okay, and its total size of the genome is 10 kb, and among 10 kb we can insert a 9 kb gene of interest. Okay, and the amount of the particles in is 1 into 10 to the power 9 titer per milliliter, and it have the ability to induce insertional mutagenesis. Okay, so that was all about the lentiviral base vector. So we have only one topics left that we will going to discuss, that is adeno associated viruses. That will be our lecture number 9. Then we will be at the end of that viral transduction lecture. So I hope this video will be helpful to you, and thank you for listening to this class.